what really attracted our attention was this article by S.K. Day's group in Cincinnati. And what they did, they look at, and this is the mouse uterine horns, and these are placental sites. That blue spots are the implantation sites here on both uterine horns, and different days of pregnancy. Mouse pregnancy is 20 days, so day 5 is implantation time. So you can identify these implantation sites by injecting blue dye into uterine horns. And what they did, that they isolated tissue from these implantation sites, which is called IS, implantation site, and the tissue between two implantation sites, inter-IS, and then characterize the tissue, isolated proteins, characterize them. What are the proteins that are identified with implantation? And this is what they saw when they analyzed the protein extract from the implantation site. These 4 proteins popped up. They were the major proteins that were associated with the implantation site, different regions of implantation site. And this is where transthyretin happened to be one of the proteins that was associated with the implantation site. This is the mouse work. We have done a first trimester placenta in humans, and we can identify this protein also being present in the decidual and placental tissue in humans. So we get back to this cartoon, as I showed you before, and basically we think that this protein could be present or might be regulating because we have isolated it, we have identified that in the placenta, and at least in mice, this group that I just talked about, they have identified this protein to be present at the implantation sites. So we started looking at some of these tissues, follicular fluid, and different stages of cell division, and then implantation sites. So let me take you through some of the work that we have completed already. This is a very preliminary work, so we still have a long way to go to complete these studies. And this is the first thing that we have done in collaboration with Dr. Jared Ravens. This is the follicular fluid from right ovary and left ovary, and these are from mature follicles. And if I remember correct, close to 18 millimeter. And we collect, and this is simple ELISA for this protein. And you can see it's present in very high amounts. These are microgram per ml concentration. So this protein is present at very high amounts in follicular fluid. And what was surprising, and this is a highly reproducible observation, the right ovary makes much more of this, or follicular fluid from right ovary has much more of this protein compared to the left ovary. We do not understand the significance of this data because in the IVF clinic, I know we don't worry about right ovary versus left ovary unless somebody can tell me what could be the significance of this type of data, but it's a very highly reproducible observation, and we always see that right ovary has much more of this protein compared to the left ovary. So this was the very first experiment. So this confirms that even at the follicular follicles level or follicular fluid level, this protein is present in human systems. So what we have done, as I showed you, the follicular fluid data is present there. We do not know whether early cell division embryos or blastocysts contain it because we are staining those tissues now with the antibodies against this protein. We know it's present at the implantation site, in the placenta, and of course in the serum. We have already done ELISA from pregnant women at different stages of pregnancy, and this protein is present in very high amounts. So this was our attempt to really go forward with this work and what we wanted to do. Does it really mean anything in real life? Would it help the embryo health in any way? And so we have tried to do here testing of transthyretin as a protein that improves embryo health and pregnancy rate. And these are some of the experimental details that I will take you through. Basically, embryos retrieved by superovulation, then they are exposed to the pregnant mare serum gonadotropins, HCG, They are mated for fertile males, and then we collect we flush out embryos. Rather, Dr. Jared Robbins' lab does that work. They flush out the embryos, and then this is how they are assigned to different experimental conditions. This SPS, serum protein substitute, so this is a global media which contains all the vitamins and nutrients, and then along with 10% of this serum protein substitute, no serum, but serum protein substitute. And we have depleted SPS of serum protein substitute because in initial experiments, we found out that this serum protein substitute contained minor amounts of transthyretin. When we did Western blotting, a lab technique that was able to detect minute amounts of this protein that was present in serum protein substitute. So we have depleted this one of that particular protein and supplemented depleted SPS with transthyretin at different doses. And this is basically the dose response. We have done at different doses, 1, 10, 100 nanogram test protein that we added to SPS, which was depleted of this protein. And of course, daily observation of embryos, blastulation rate, hatching rate, arrest rate. These are the experimental details that we followed. And as I said in the previous slide, blastulation rate, hatching rate, arrest rate, and total cell number calculated on PC day 4.5. So we put on day 1, and then we followed them for 4.5 days. And then just to count the cells, we co-stain with these chemicals, propydium iodide and bisbenzamide. And I will show you a picture how it looks. We mount this embryo in glycerol, and then we do serial Z-stack confocal microscopy to see. And this is the kind of thing, if this protein is helping the growth or cell division, that this is what you would see. These red cells are the trophoectoderm cells, and this is the inner mass here. So those two dyes that I talked about, propydium iodide and bisbenzamide, they will stain trophoectoderm cells and inner mass cells, and this is what you see. You can count these cells and calculate whether this protein is helping cell division or not. And these are the very preliminary data that we have come up with. Here, it does help if you deplete the SPS of this particular protein and then supplement that one with this protein at 10 nanogram per ml. You do get back the blastulation is improved in response to this protein. Look at hatching. This is the protein depleted of this, the mixture depleted of this protein, but when you add it back, you improve hatching a great deal. Similarly, the growth arrest is there, observed in response to depleted mixture, but when we add this protein back to this here, we can inhibit the growth arrest. So according to these data, we can say that this protein is helping hatching, and it's also inhibiting the growth arrest that takes place in many of the embryos, that they may reach 2-cell or 4-cell or 8-cell stage, but then they don't divide further. And this is what leads to the arrested growth, and this protein can improve this situation. So we think that this protein would be very helpful in terms of enhancing hatching and inhibiting the growth arrest. And we do not have enough zygotes that we have used at this point to really say that there's a statistical significance for blastulation. So we are working on that. We continue to do work and add more zygotes to these studies. These are all experimental data in mice. We are not allowed to do these things in humans at that point. So once we confirm all these things in this model, then we'll move on to the human models if we are allowed to do some of these things. So these are some of the data that we have accumulated so far. We do not know yet whether it will result in better pregnancy rates. And these are the experiments currently being performed in the lab, where we will take the, where we'll take these blastocysts and implant them and see if it improves the pregnancy rate. And those experiments are currently being done, and we have no information at that. And that's the critical point. If we cannot improve pregnancy rate, then it really doesn't mean much. So this is what we are really focusing right now at this point, to complete these experiments to see whether this process will help in improving pregnancy rates. And if that happens, I think that will be an impetus to take this work to the human settings. So, as I said, this work is still in progress. In the meantime, we conclude that transthyretin is present at high levels in mature follicles. This protein improves blast formation and hatching. And this protein also significantly reverses growth arrest during cell division. We propose that embryo culture medium should contain adequate amount of this protein, transthyretin, for better growth and health of the embryo. And pregnancy rates should provide better insights for its clinical use. And that is the main focus of when we can demonstrate that this protein will improve pregnancy rates, and that would be an impetus for us to really take it to the next step for its clinical use. So let me just acknowledge the people who have done the work. This is from my lab, Dr. Kalkunte and Wendy Norris. They are the main people who have helped in this work. And as I said, this work has been done in collaboration with Dr. Robbins' group by Laurie Underhill, who is a postdoctoral fellow in Dr. Robbins' group. And these are some of the other people in the lab. And of course, this work won't be possible without the support from these funding sources here. So thank you very much.
So no, you said like there is a lot of amount in the follicular fluid, and in fertilization it is helping. So means the follicle is released, and normally the fertilization is happening at the tubal level. So in the normal cases, how is it going to affect?
So basically what it is that we personally feel that even the embryo, when it's developing, it's producing this protein. So you have the follicles produce it, and then the embryos will also produce the embryonic cells. The trophoectoderm particularly will produce this protein and help the cell division. So that's one way. So that would be the autocrine factor activity. And if it is really ferried on from the follicular fluid, and we don't know about the seminal fluid, whether seminal fluid would contain this protein or not. So that might be further carried on and through the fallopian tube, but we do not have any data at this point on that.
Another thing is, like normally you said right side produces more transthyretin than the left. What my take is, you should study in those cases where the right ovary is missing, whether there is a compensatory increase in the left side. In such cases,
excellent point. We have discussed with Dr. Robbins already, although we did not. There are some controversial articles in the literature suggesting that you get better implantation using the egg from right ovary or left ovary, but they are highly controversial, and we have talked about those articles, and we do not think the information is statistically significant in those articles. So if anybody has a bright idea about the differences between right ovary and left ovary, we would like to really hear about those ideas. If someone can tell us the why right ovary versus left ovary, I mean, there's a difference in terms of production of this protein.
The right ovary and the left ovary.
Say it again.
Venous drainage, sir. The ovarian vein, it directly empties into the vena cava on the right side, and the left side through the renal vein. So maybe that you said this protein is related to this dialysis and all. Somehow it's related to the kidney and all.
Yeah, those discussions have taken place. Then when we discuss about, when we saw this data, I questioned Dr. Robbins, what do you think about it? He said, in the IVF clinic, we don't worry about right versus left ovary. So what is the significance of this protein being produced in much higher quantities by right ovary is still a big question mark, and we can always speculate that this could be an explanation, but we really do not have any concrete idea at this point.